Hello everyone, welcome to the Swayam course, Genetic Engineering and Recent Advancement. I am Dr. Richa Sahu, working as an assistant professor at MIT Institute of Biotechnology, MIT University, Chhattisgarh. So, till now in this course we have talked about the basic concepts of genetic engineering, the basic techniques that are involved in genetic engineering, the vectors that are used. We have talked about DNA replication, we have talked about gene expression, gene regulation. So, today we will be talking about reporter genes. Ultimately, one wishes to determine how genes and the proteins they encode function in the intact organism. Although it might sound counterintuitive, one of the most direct ways to find out what gene does is to see what happens to the organism when the gene is missing or it is absent. This is done by mutating the gene or removing the gene. So, another way to understand the gene function is you can get the clues of gene function often by obtaining, or it can be obtained by examining when and where a gene is expressed in the cell or in the whole organism. Determining the pattern and timing of gene expression can be accompanied by replacing the coding portion of the gene under study with a reporter gene. So, before starting the reporter gene, let us understand what exactly it is, in what category it belongs to. So, basically let us talk about the marker gene first. So, marker systems or marker genes are the tools used for studying the transfer of gene into an experimental organism. In gene transfer studies, a foreign gene called as transgene is introduced into an organism in a process called as transformation. The common problem for researcher is to determine quickly and easily if the target cells of the organisms have actually taken up the transgene or not. The marker allows the researcher to determine whether the transgene have been transferred in the test organism, where it is located and when it is expressed. So, marker genes or the marker systems exist in two broad categories, the reporter genes and the second one is the selectable markers. Talking about selectable marker gene, they are basically these genes are usually an integral part of plant's transformation system. They are present in the vector along with the target gene. In a majority of cases, the selection is based on the survival of the transformed cell when grown in a medium containing a toxic substance, like for example antibiotic, herbicide or antimetabolite. This is due to the fact that selectable marker gene confers resistance to the toxicity in the transformed cells, while the non-transformed cells get killed. An example of this is antibiotic resistance gene, where then antibiotic resistance gene is along with the vector is transferred or transformed into the cell, and they are grown in the presence of that particular antibiotic. The bacteria is grown in the presence of that particular antibiotic. So, the transformed cells who have integrated this antibiotic resistance gene will only be able to survive with respect to the other cells which do not have this antibiotic resistance gene. So, this is one example of a selectable marker gene. So, talking about the topic that we are going to discuss today is the reporter gene. So, a reporter gene is a gene which produces a specific phenotype, in turn enabling the differentiation of the cells processing this particular gene from the cells with which do not have this particular reporter gene. Hence, the transformed cells can be selected from the thousands of non-transformed cells on the basis of this particular reporter gene. So, the reporter genes form specific protein products which are easily detectable and quantifiable. Reporter genes are the invaluable tools to track and to study another associated gene in bacteria and in mammalian cell culture or in animals or plants. A reporter gene is an exogenous coding region joined to a promoter sequence or element in an expression vector that is introduced into the cells to provide the means of measuring the promoter activity. basically the transcription, that is RNA synthesis and then protein synthesis. Once expressed in cells, the reporter gene or the reporter is assayed by either directly measuring the reporter protein itself or by assessing the enzymatic activity, correlating the strength of the fused promoter element with the amount of reporter produced. The ideal reporter gene is not endogenously expressed in the cells of interest and amenable to assays that are sensitive, quantitative, rapid, easy, accurate, reproducible and safe. This is the image of a typical gene construct which has a reporter gene. In most the cases, the expression of the reporter gene is then monitored by tracking the fluorescence or bioluminescence or enzymatic activity of the protein product of the reporter gene. Here you can see the green part is the reporter gene along with the promoter region in the DNA or the gene construct is synthesized at the particular mRNA, and finally the reporter protein is synthesized because of the reporter gene that is present, and the amount of the reporter protein or the reporter product is directly proportional to the the amount of gene that is expressed. So, gene expression is controlled by regulatory DNA sequence located upstream or downstream of the coding region, which are not genetically transcribed. These regulatory sequences which control which cells will express a gene and under what condition can also be made to drive the expression of the reporter gene. One simply replaces the target gene coding sequence with that of the reporter gene and introduces these recombinant DNA molecules into the cells. The level, timing and the cell specificity of reporter protein production reflect the action of the regulatory sequence of the regulatory gene that belong to the original gene. Here are some key features that we will be talking about the reporter gene. What should be the key features of a reporter gene? When can you take a particular gene or a particular sequence or a particular phenotype as a reporter gene? The first you can see the feature should be the visible and detectable output. Reporter gene produce a visible or easily detectable output such as the color change, the fluorescence or bioluminescence that can be measured and quantified directly. Next is non-interfering. Reporter gene should not be interfering or should not interfere with the normal function of the host cell or the gene being studied. They should have minimal impact on the biological process of interest. Next feature should be stable expression. Reporter gene expression should be stable over time, allowing for long term experiment and measurement of the particular reporter gene. Next is selectable marker. In some cases, reporter gene can serve as a selectable marker, allowing cell to express the reporter gene to be easily distinguished and selected from those who do not have that particular gene. Next is compatible promoters. Reporter gene can be driven by the variety of promoters including constitutive, inducible or tissue specific promoters depending on the experimental goal that has been set. Next, a very important feature for a reporter gene is quantifiable. The reporter gene output should be quantifiable, allowing for accurate measurement of gene expression levels. This can be achieved through the use of appropriate instrument and assays. There are various variants of reporter gene that can be used. Different reporter gene can be used for different applications. Common reporter gene include beta galactosidase or known as LAC-Z, green fluorescent protein or GFP, luciferase, alkaline phosphatase, etc. Researchers choose the reporter gene based on the specific requirement of the experiment. The next feature is there should be minimal or minimum crosstalk. Reporter gene should not crosstalk with the endogenous gene or other reporter gene in the same experiment, which can easily mislead the result or that can lead to false results. The integration of the reporter gene should be stable as mentioned earlier in the previous slide. In some cases, reporter gene can be stably integrated into the host genome to ensure their presence in all the daughter cells during the cell division. The cell type specificity. Depending on the research goals, reporter gene can be selected to provide cell type specificity or cell type specific expression patterns. This helps researchers to study gene expression in a specific cell type or tissue. Another is for in vivo in vitro applications. A reporter gene can be used in both in vivo, that is in living organism, and in vitro, that is cell culture experiments, making them a versatile tool for various research settings and various research experiments. Next is the temporal control. Some reporter gene can be used to study temporal changes in gene expression by using inducible promoters or degradation signals. So, these were the key features of a typical reporter gene, or these are the things that has to be kept in mind when the reporter gene are selected. So, now we will talk about the examples, some common examples of reporter genes. The first is the gene that encodes jellyfish green fluorescent protein or GFP, which causes cells that expresses it to glow green under UV light. The first example is GFP or green fluorescent protein. The next is the enzyme luciferase, which catalyzes a reaction with luciferin to produce light. Next example, another common reporter gene which is used in bacteria is the LAC-Z gene, which encodes the protein or enzyme beta galactosidase. What happens is this enzyme causes bacteria expressing the gene to appear blue in color when grown on a medium that contains a substrate analog X-gal. So, this is how if the bacteria is producing blue color colonies, that means that particular reporter gene is expressing. An example of another selectable marker reporter in bacteria is the chloramphenicol acetyl transferase or CAT gene, which confers resistance to the antibiotic chloramphenicol. So, how reporter genes actually work? So, reporter genes and their associated detection method have contributed greatly to the study of the eukaryotic gene expression and regulation. Although reporter genes have played a significant role in numerous applications both in vitro and in vivo, they are the most frequently used indicators of transcriptional or translational activity in the cells. But how do they work? The reporter vectors that is plasmid that contain the reporter gene are used to identify and characterize promoter and enhancer element function, because reporter protein expression corresponds with the transcriptional activity of the reporter gene. For these type of studies, promoter sequences are cloned upstream or downstream of the gene. The promoter reporter gene fusion is introduced into the cultured cell by standard transfection methods or into the germ cell to produce transgenic organisms. Thus, you can characterize promoter and enhancer elements that regulate cell, tissue and development defined gene expression by using these reporter genes. And how do you measure the reporter gene activity? So, the reporter gene, or rather the protein translated from the reporter gene sequence, can be assayed by detecting enzymatic activity or spectrophotometric characteristics or indirectly with antibody based assays. There are numerous assays that can be used to measure the reporter gene activity. So, in general, enzymatic assays are more sensitive due to the small amount of a reporter enzyme required to generate detectable levels of reaction products. A potential limitation, however, of enzymatic assays is high background if there is an endogenous enzymatic activity in the cell. For example, beta galactosidase break down lactose. Antibody based assays are generally less sensitive, but will detect the reporter protein whether it is enzymatically active or not. So, how is, as I mentioned, how actually it is measured? Fundamentally, a reporter assay uses a protein to measure biological outcome using an observable parameter like bioluminescence or chemiluminescence. On the basis of bioluminescence or chemiluminescence, you can measure the activity of the reporter gene. Let's talk about the examples one by one. We will start with green fluorescent protein or GFP, one example of a reporter gene. So, green fluorescent protein or GFP is widely used reporter gene in molecular biology and cell biology research. So, it was first discovered in the jellyfish Aquaria victoria, if I am not wrong, and has since become a valuable tool for studying the gene expression, protein localization and various cellular processes. So, what does it do? So, fluorescence, it basically emit fluorescence or basically produces fluorescence. So, GFP or green fluorescent protein emit green fluorescence when exposed to ultraviolet rays or blue light. This property allows researchers to visualize and track the expression and localization of GFP tagged protein in living cells and the organisms without the need for destructive techniques like staining or fixation. So, if the GFP protein or the reporter gene is expressing, it will glow or it will exhibit fluorescence in the presence of UV light or blue light. So, GFP is used as a reporter gene because its expression is directly linked to the activity of the promoter or regulator element of interest. When a promoter or enhancer sequence is placed upstream of the GFP gene in a DNA construct or a genome construct or a gene construct that you are using, the level of GFP fluorescence serves as an indicator of the promoter activity. More the GFP fluorescence, more is the activity of the promoter. So, this is how you quantify or this is how you measure the activity of the reporter gene, and indirectly or directly you measure the promoter's activity. In this image you can see the cells, on the left hand side it is C. elegans, on the right hand side it is the mice. Both are expressing the reporter gene green fluorescence protein. You can see the bioluminescence or the fluorescence, not the bioluminescence, the fluorescence, the green fluorescence of this is which is the indicator of the expression of the GFP reporter gene, which is which you can see that green colored in the presence of UV light or blue light. This is how the GFP is expressed as a reporter gene. So, what are the applications of GFP? Where are they used in genetic engineering? They are used for the study of gene expression. GFP is often used to study gene expression patterns in cells and tissues. By fusing GFP to a gene of interest, researchers can monitor when and where the gene is turned on or off. Also, the next very important application of GFP is, as I was showing in the image just in the previous slide, is protein localization. GFP can be fused to a protein of interest to track its subcellular localization. This is specially useful for studying dynamic processes within the cells. Next is the protein-protein interaction. Split GFP assays enable the visualization of protein-protein interaction by tagging interacting protein with complementary fragments of GFP. As I have mentioned previously as well, there are different variants of reporter gene. In a similar way, there are different variants of green fluorescent protein as a reporter gene. Each variant of GFP have a different spectral properties, such as enhanced GFP known as eGFP, cyan fluorescent protein CFP, yellow fluorescent protein YFP and red fluorescent protein RFP. These variants allow researcher to label multiple proteins simultaneously and study complex biological process. For example, all these variants can be used together to tag different different proteins or different gene, so that when they are expressed when they are expressed together, different colors or different fluorescent colors will show the the localization of the protein or the expression of the gene. Next is category is the bioluminescent reporter gene. So, what is bioluminescence? It is different from fluorescence, like it is different from GFP. So, bioluminescence is a special form of chemiluminescence found in living organism. This class of chemiluminescence is basically luminescence or glow because of a chemical reaction. So, in case of bioluminescence is because of the biological reaction. So, this class of chemiluminescence is an enzyme catalyzed process where the high efficiency of photon emissions is derived through natural evolution. The enzyme are called luciferases and the photon emitting substrates are the luciferins. Bioluminescent chemistries have evolved from multiple independent origins and comprise many distinct molecular structures. Of many natural forms, two have been widely and extensively used for as a genetic reporters assay, that is firefly luciferase and Renilla luciferase. These are the two examples of the bioluminescent reporter genes or enzymes. How do they work and what is the advantage of bioluminescence over fluorescence? So, bioluminescence reporter gene assays have distinct advantage over fluorescent assays like GFP or green fluorescent protein, in that they can deliver 10 to 1000 fold higher assay sensitivity in comparison to GFP. What happens is this greatly increases sensitivity and substantially improve assay performance, and when applied to complex biological samples. The reason for this becomes clear when you examine the mechanism of light produced by fluorescence and luminescence. Both processes yield photons as a consequence of energy transition from excited molecular state to the lower energy orbitals. However, they differ in how the excited state orbitals are created. In case of luminescence, the excited states are the product of exothermic chemical reactions, whereas in fluorescence the excited state are created by absorption of light. Please understand, in case of luminescence the excited state are generated as a product of exothermic chemical reactions. In fluorescence, however, the excited state are created by absorption of light. In reporter assays, bioluminescence has the advantage because photons are not required to create excited state. Therefore, they do not constitute an inherent background which when measuring photon efflux or efflux from the sample. The resulting low background permits precise measurement of small changes in the light across 4 to 8 orders of magnitude in linear range of the assay. That means, if there is a because the background is dark, the background remains dull, that is why a little bit bioluminescence can be detected in comparison to the fluorescence, because in fluorescence the background is also very dark, very luminate, very bright. So, you cannot detect very small amount of changes in the light in comparison to the bioluminescence. The example of bioluminescence reporters is luciferase reporters. The luciferase reporter technology is based on the interaction of the enzyme luciferase with the luminescent substrate luciferin, which releases light by the process of bioluminescence. Bioluminescence is found in a number of different organisms. However, it is the firefly luciferase that is by far the most commonly used bioluminescent reporter due to both sensitivity and convenience of the enzyme assay and to the tight coupling of the protein synthesis with enzyme activity. What happens in luciferase reporter, or what happens if you are using luciferase enzyme as a reporter enzyme? The monomeric enzyme of 61 kilodalton luciferase catalyzes a two-step oxidation reaction to yield light, usually in the green to yellow region, typically 550 to 570 nanometer wavelength. Upon mixing with the substrate, firefly luciferase produces an initial burst of light that decays over about 15 seconds to a low level of sustained luminescence. The gene encoding firefly luciferase Luc gene is a cDNA and does not require any post translational modification. This means it is available as a mature enzyme directly upon translation from its mRNA. It is an important consideration when using a reporter gene outside its naive appointment. This is one of the advantage of this Luke gene. So, this is the image how bioluminescent or luciferase enzyme reporter gene works. You can see the different colors, you can see how they are glowing in the dark background. This is how the assay looks when you are using luciferase enzyme as a reporter enzyme. The next example that we are talking about is the LAC-Z reporter gene. A LAC-Z reporter gene is a commonly used genetic tool in molecular biology and microbiology. And how does it work? It is derived from the LAC operon of Escherichia coli or E. coli, and it is used to study gene expression and regulation in various organisms, not just in E. coli, but in various organisms and various hosts. The LAC-Z gene encodes beta galactosidase enzyme, an enzyme that breaks down lactose into glucose and galactose. How this LAC-Z reporter gene works as a reporter gene or to study the gene expression? So, the promoter region, let us talk about the promoter region. In molecular biology experiment, a researcher will often want to study the activity of a specific promoter, which is DNA region that controls the initiation of gene transcription. The LAC-Z reporter gene is inserted downstream to the promoter region that the researcher or the person working is interested in studying. So, whatever gene or whatever promoter region you want to study, you insert this particular reporter gene downstream of it. Now, how the LAC-Z gene is expressed? If the promoter is active and allows transcription to occur, the RNA polymerase enzyme will bind to the promoter and initiate the transcription of the LAC-Z gene. This results in the production of beta galactosidase enzyme in the cell. And how this beta galactosidase enzyme activity works? Beta galactosidase is the enzyme that can cleave a substrate called XGAL, which is 5-bromo 4-chloro 3-indolyl beta D galactopyranoside. When XGAL is cleaved by beta galactosidase, it produces a blue colored compound. Therefore, if the LAC-Z gene is expressed, that means it is transcribed or translated, due to the activity of the promoter being studied, the cells containing the reporter gene will turn blue. The colonies of the E. coli, whatever bacterial or cell that you are using, that will appear blue colored. So, that is why it is used as a reporter gene, because the colonies turn blue and you can easily recognize whether the gene has been or the promoter region is expressing or not. So, this completes the reporter gene. This lecture gives you an overview of reporter gene, what are reporter gene, what category they belong to, and why they are known as reporter gene. So, in this lecture we talked about the basic concepts of a marker gene or the marker system under which the reporter gene comes. We talked about what is reporter gene and how they work and where they are inserted in the gene construct. Also, we talked about the applications of the reporter gene in genetic engineering. We talked about the 4 types of the 4 examples of reporter gene, namely the GFP, the luciferase, the XGAL, and one more. And in detail we talked about the 3 types of reporter gene which are primarily used in genetic engineering, that is green fluorescent protein which exhibits fluorescence. Then we talked about bioluminescence, where we talked about luciferase enzyme and how they work as a reporter gene, and finally we talked about XGAL gene as a reporter gene. So, in the next lecture you will talk more about the concept of genetic engineering. Thank you very much.